Welcome everyone to BioGenerator. I'm Jim McCarter, Senior Managing Director, and BioGenerator Ventures creates, builds, and invests in companies that are solving important problems in human health and agriculture. We're a program of BioSTL that has invested nearly $50 million to help start 134 companies in St. Louis over the last 15 years. And those companies have gone on to raise an additional $2.8 billion in follow-on funding. We've supported 170 companies with space in the BioGenerator labs and the Helix Center, and hundreds more with mentoring of entrepreneurs and in residence and experts in SBIR grant writing. So this is our first Innovators Hour at BioGenerator, and I'm thrilled to have the opportunity to speak to Dr. Christian Harding, the co-founder and chief scientific officer of Omniose, a St. Louis-based company that is revolutionizing vaccine development. Omniose is a BioGenerator portfolio company and is headquartered here in the BioGenerator labs at 4340 Duncan. And I think you're currently the largest company in the labs, right? With something like...
I'm going to defer to you.
I think we can ask Sophia, but I think over 20 people or so that are on.
Yeah, we're going to be, I think it's 18 here, 19 total. We have one remote CEO in Boston. Right. So yeah, big footprint here.
Excellent, excellent. And so Christian, just to get us started, we'd love to have you kind of tell your life story and entrepreneurial journey. And so I know you're from South Carolina and you chose Ohio State for grad school. So tell me the story of kind of how you got started in your PhD work, how you know, what led you to that PhD work, and then how you met Mario Feldman, who's now at Washington University.
Yeah, so first of all, thanks for the invitation. It's great to see so many faces out here. I went to a small liberal arts school, College of Charleston in South Carolina. Best time of my life. Go to Charleston, South Carolina, visit. Loved biology, so thought I wanted to go to med school, didn't know. Waffled a little bit, you know, took a year off to figure out, do I really want to do med school? I was the chem stockroom manager. I set up OChem lab for everyone. I realized I didn't want to go to med school, applied to graduate schools, just umbrella programs in general. I applied to 3, University of Wisconsin, I got rejected. Wash U, I got an interview. I didn't get accepted, so I became a proud Buckeye. Got accepted into Ohio State, had a great experience, did multiple rotations across fields. I'm a bacteriologist currently, but did rotations in epigenetics, neurobiology, cardiovascular disease. I joined the micro lab, Dr. Robert Munson's lab, because the people were nice. And I figured if I was going to be there for 5 years, it was advice I got going into grad school. Make sure you really like the people you work with. And I liked the people, and so I became a bacteriologist. And during that experience, I was technically at Nationwide Children's Hospital. It's the pediatric affiliation for Ohio State University. I started collaborating and studying some weird bacteria called Acinetobacter. And my thesis program kept going further and further, and it was getting out of my realm of comfortability. And so I reached out to an expert in the field, Mario Feldman, who's our co-founder of Omniose. He was at the University of Alberta. We worked together for a year and a half, and he offered me a job. I said, no, thank you. My wife will kill me if we move that far north. And he said, lucky, I just got recruited to Wash U. Will you join me? And I joined him. Spent about a year and a half there, and then spun the company out subsequently.
Great, so let's spend a little bit of time on the technology. And I've been trying to kind of get up to speed on understanding enough immunology to understand what you're doing. But maybe a good place to start would be to tell the story of why are conjugated vaccines so important? Because I understand that stopping bacterial meningitis, over 1 million people that used to die every year are no longer dying because of conjugated vaccines. And so tell me, how does that technology work, and why do you have to conjugate the carbohydrate to the protein?
Yeah, so we, in a good way, take for granted bacterial vaccines because they're so effective. They work so well. You don't have to get annual boosters. And that is largely due to conjugated vaccines, glycoconjugate vaccines. It's all the same name for a different name for the same thing. Bacteria are covered in a sugary coating. It's very unique. It's like a fingerprint. That's great for teaching your immune system. It looks different than you and me. The problem is those carbohydrates, they're very poor vaccine antigens. They don't give a memory response. They're not good at creating IgGs or antibodies that are good at effectively clearing the bacteria. So about 50 years ago, a group discovered that if you conjugate that bacterial antigen to an immunogenic protein like diphtheria toxoid or tetanus toxoid vaccines that work very effectively, you can teach the immune system to recognize these bacterial antigens. Hence, conjugate vaccines are born. Late 80s, early 90s, the first conjugate vaccines are licensed, and since then, we've seen multiple pneumococcal conjugate vaccines, Neisseria meningitis for meningococcus, and they work remarkably well, but they're exceedingly difficult to manufacture. They're one of the hardest drugs.
Before we go into the manufacturing, so just staying on the science of, so if you have the carbohydrate alone, even with the protein but not conjugated, it doesn't, the MHC class 2
Yes.
doesn't display it, or what,
So
why is that?
immunologically speaking, yes. Your B cell is going to try to show that to a T cell, and they're going to have an interaction to teach each other. The carbohydrate antigens don't bind that pocket. It's called MHC class 2. So a B cell can't show a T cell and therefore be taught to reinforce that interaction and make a mature antibody response. So they're classically called T cell independent antigens. But if you couple it to a protein,
Right.
that protein can bind that MHC class 2 cleft and be presented to your T cells or other immune cells. Hence, you can get that T cell dependent antigen and get a nice tight interaction between those and affinity maturation. All the good things that we take advantage of in monoclonal antibody development, the conjugate vaccines now allow you to get that polysaccharide specific response, that bacterial sugary specific response, and get a very effective immune response.
Right. And then, so to follow on with where you were headed, which was toward the manufacturing, so that prior to your work, that generally the way these have been manufactured, rather than manufacturing them biologically in E. coli, it was to actually do a chemical conjugation. And so maybe tell us a little bit about, why is that so difficult, and why is that such a barrier to commercialization of these vaccines?
All conjugate vaccines are glycoproteins. It's a polysaccharide or carbohydrate attached to a protein. There are a lot of ways to make glycoproteins. The prevailing early manufacturing processes did semi-synthetic, extract, purify that antigen, so grow the bacteria in large fermentation, thousands and thousands of liters, extract it, and then chemically link it to a purified protein. It is a tried and true technology. It works. It's just very difficult to scale because you have to be careful not to modify that bacterial fingerprint in any way. Your immune system is very smart, and these chemical processes, these chemical conjugations, inherently have to augment that bacterial fingerprint to enable the protein and polysaccharide to bond. So there's...
And you mentioned one took 2 years to manufacture?
Yeah, Prevnar 13. Now we're up to Prevnar 20. So Pfizer's wonderful vaccine. Yeah. But yeah, Prevnar 13 in 2015, one dose took 2.5 years to manufacture from start to finish. Wow. Also, it's 13 vaccines in one tube. Again, so it's 13 different types of conjugate vaccine in one tube in a single immunization.
Got it. So we've kind of talked a little bit about the science of kind of why this is important, set the stage kind of then for Omniose. So you're here in St. Louis working with Mario. And so then what happened that led to the formation of the company?
So I realized I didn't want to be a professor, was number one. It's hard for all the postdocs out there, graduate students. It's a lot of pressure and long hours and not great pay. And I got burned out quickly, 1.5 years in. And so I went into Mario's office. I knew we had a technology, and I'm ready to do something different. I was like, do I go to pharma? What do I do? And he's like, let's open a company. He had a history of that. He had other companies that he was affiliated with throughout his academic career. And so he had some experience. He knew a lot about patents. He had multiple things that he had licensed to GSK. So I was fortunate. I got a little bit of a head start because I was with someone who knew. Unfortunately, we didn't know a lot about starting companies from, like, let's call it ground zero. We had an idea, a technology, proof of principle. Where do you go next? So we stumbled around. We talked to this person, that person. Eventually, we ran into a company over at the old BioGenerator facility in 20 South Sarah. And they heard our idea and they said, you've got to talk to this guy, Harry Arator. Many of us in here, Harry Arator's near and dear to our heart. And he played a pivotal role in helping us in some way. So we got introduced to Harry. We pitched him on the idea of what we want to do, how we want to transform manufacturing conjugate vaccines using our process. And he was sold. He's like, I will help you. I will help you write a grant, an SBIR, STTR business grant.
And so this was July 17th of 2016, and the grant deadline was September 5th.
Yes.
You didn't.
I'll help you.
And you didn't have a company yet.
Yeah, the company formed August 4th of that year.
Okay.
So he said, I'll help you, but we'll do the January 5th submission. We'll get there. And I think it was probably just ignorance. We didn't know what we were getting into, that we just really pressured him. We can do this, we can do this. And so he relented and he helped us get all of the registrations. And so I just wrote and just worked for 6 weeks and we got it in. And then you wait a while, a couple months.
Yeah.
But I called Harry and I said, I got a score. I don't know what it means. I got a 17. And he was like, what? That's really good. I was like, can you help me with another one? And so we did 2 more back to back, and those were all funded.
And now you've received over 12 million in SBIR funding.
Been awarded, some of them yes. Some of it is in multi-year grants, but yeah, we're at 11.8 million with another 3 million pending. So happy to talk grants in the next session if anybody wants to. So grantsmanship is a game.
Yeah, so maybe say a little bit for the budding entrepreneurs about the importance of non-dilutive funding.
I mean, all funding has strings. Even non-dilutive funding has strings. It's called the Bayh-Dole Act. There's a lot of reporting, right? There's iEdison, right? There's a lot. It's non-dilutive. It doesn't, dilute out equity in your company, but there are strings. But I think the power is that we should all recognize and think of the NIH as pre-seed money. Even at the R01 level, right, with the basic research grants, that's how I think about it. Especially if you're in an area that is not inherently hot. I don't work on oncology. I don't. Oncology is inherently much hotter than bacterial vaccines. So non-dilutive funding, SBIR grants, can really float early companies if you're maybe not in the hottest area. Specifically when you look at a group like the NIAID, whose budget is as big as NCI, and they're congressionally mandated to give out almost as much money as NCI. So there's a lot of funding opportunities out there if you have an idea.
Yeah. So let's go back into the science for a minute, because I didn't really ask you about the company was originally VaxNumo,
yes,
and you
correct.
Renamed it Omniose.
Yes.
Tell a little bit about, because I think the name Omniose kind of explains what the oligosaccharide transferase does and why it's so game-changing.
Yeah, so our technology, we make glycoproteins. That's what a conjugate vaccine is. So as I said, there's lots of ways to make glycoproteins. So the way that we do it is entirely biological. So rather than relying on some chemistry or full chemistry, we rely on enzymes to do everything for us. So E. coli is lab-safe bacteria. It's the workhorse of recombinant biology. More than 30%, or approximately 30% of all FDA biologics are still made in E. coli. So we've tuned and taught E. coli to make the bacterial fingerprint for pathogen X, bacteria X. We simultaneously taught it to make a carrier protein, that immunogenic body that's required for that conjugate vaccine. And we have developed a technology, an oligosaccharide transferase or a conjugating enzyme, that couples the polysaccharide to the protein. So instead of relying on chemistries to do this to that, we rely on an enzyme to do it. We call it bioconjugation. So that process.
And then Omniose means?
We started Vaxnumo. We were exclusively doing pneumococcal conjugate vaccines, so pneumo, we were just trying to do a play on words. That's what happens when you form a company in an office in like one day. Your name is not that good. Vaxnumo, I admit it, it was bad.
So it doesn't have to do with Missouri?
Yeah, well, it was everything. It was New Missouri, Newco in Missouri. It sounded like pneumococcus, but it didn't stick. So the power of our platform is, I actually didn't invent bioconjugation, but Mario, my postdoc advisor, did when he was a postdoc in Switzerland in 2005. And they spun out companies associated with that, and GSK ultimately acquired that. That first bioconjugation company had limitations, very powerful, multiple vaccines in clinical trial, but it had limitations on what polysaccharides it can transfer. Our technology, bioconjugation 2.0 for a lack of a better term, is more universal. It can transfer any or all sugars to any or all proteins. So omni just meaning everything or all, and ose just being the suffix for sugar, like glucose, mannose. So Omniose was born out of, we needed a new name, and it was more apt as to what we do.
Got it. And so I want to talk a little bit about how you've worked with BioGenerator. So BioGenerator provided funding, provided lab space, but Harry's help on the grants, but there are a number of other ways in which you worked with BioGenerator.
So I-6 grant funding. So some people in here probably remember I-6 grants. Those are great. Those were used to, I was awarded those, and they were used to pay patent expenses. We were at a national stage, a point where we were filing nationally, and grants can't be used for intellectual property purchases or expenses. So I-6 was enormous, those funds that BioGenerator helped us with. Executive searches with Alan and Harry as well to recruit and land a former chairman, extensively network, negotiating a royalty agreement with the University of Alberta. Our technology is legally a spin out of the University of Alberta because that's where Mario was when we co-invented the technology. And so that was a very arduous, difficult process. And we didn't have a chairman or an official CEO at the time. So Harry and BioGenerator were instrumental in that negotiation. And then just letting us grow organically. I think we take for granted that.
And I heard that, was it true that you were actually literally in a closet at some point?
Oh yeah, who remembers that? Who was over? All right, Crystal does, Mike does. It was free, so it was right across the street. One of those caveats about, which is a good caveat, like you can, before the feds will give you the money, they want to see that you have a lease. It's probably smart. And so I had to BioGenerator gave me a lease for space, but there was no space. So I had an address, and it's where the nanostring was or whatever too. Canopy, they had equipment in there too. And so I was glad to get out, when I got out. So that's where it started, but it was like a utility closet. Yeah. So fond memories, we've grown, thankfully.
And today we've got 76 companies where BioGenerator Labs is their address. 30 of those have active experiments and bench space and are active in the lab. But many of those other companies, it is an address while awaiting funding and awaiting to grow.
Yeah, de-risking.
Yeah. So then, jumping ahead a little bit, tell me a little bit about recruiting. Your CEO is named Tim Cook, which is not Apple Tim Cook, but different Tim Cook.
Yeah.
But tell a little bit about how he came on the scene, and then I'd love to hear the story of the World Vaccine Congress in Washington in April of 2022 and what you were doing to get ready, and then what happened there.
Yeah. So I met Tim. Harry actually provided the original introduction through just going through his network, landed on Tim, met him probably in 2018, 2019, it's pre-COVID. we liked each other. We did some contract work. He would review, I think, the commercialization plan that Veronica wrote for us. So other shout out, there was also grant writing that BioGenerator helped us with too in Veronica's company. So he reviewed it. We liked each other, stayed in touch over the years, and found that information is currency. I wanted him to be the CEO, but it just wasn't the right time for him. He had other opportunities. We were still a little too new. So just kept giving updates, semi-confidential to make it juicy.
Sure.
Eventually, Alan recircled back on him in the executive search. We landed on Roman as a chairman, but the timing just worked out that he could join. His company was bought, the company he was with, and came on as a part-time CEO. We realize Vaxnumo is not the best name and undergo a rebranding, Elasticity, a local firm. I recommend them as well. Came up with Omniose, going to go out on the tour now. You want to go to your big conference, whatever that is, and announce, you've got a new CEO and a new chairman and a new name. Yep. And that's what we did. We went to the World Vaccine Congress. It's annual in DC every year. And we do that, press release. Tim has been in the industry for 30 years, so in my field it's pretty small, so everyone knows Tim. That's a plus. And during the event, he's networking, and I'm riding his coattails, and he finds a wallet on the ground, opens it, and it's somebody he knows, and he returns the wallet to them. It's a friend of his at AstraZeneca, and he's like, "I found your wallet," and he's like, "Well, thanks, Tim." He's like, "By the way, what are you up to?" He's like, "Thanks for asking. I'm actually the CEO of this company called Omniose. We're developing polysaccharide protein conjugate vaccines." And he goes, "We might be interested in that." And that's what started the negotiations of just figuring out a life to form a strategic partnership with AstraZeneca. The science was the same. Sometimes it's just the right network or just the right person knowing, connecting, because all pharma maybe aren't going to tell you what their needs are. They're going to scout. I was just very fortunate to be in an opportunity.
Luck favors the prepared.
Yes.
In that the technology was ready to go. It was primed for an opportunity.
Inadvertently operating as a stealth mode company for 6 years because I was unsuccessful at raising, let's call it, blue chip VC, obviously BioGenerator contributing and lots of grants, but grants aren't as flashy and sexy as a Series A or B, C. So we were given enough time to mature the technology so that when the opportunity did present itself to show off to big pharma, we were more than ready and landed a deal ultimately because of that preparedness.
Excellent. So we have about 10 minutes for questions, and I'll repeat the questions so that we capture them so that everyone can hear and we capture it on video. But before we jump into the questions, let me just thank Christian for taking the time to do this with us, and thank you all for coming.
Thank you.
So, Dave.
So what does the company look like in 5 years commercially? You've talked about starting a company and growing 20, very successful in the sense of a startup. But what does it look like in 5 years commercially?
So the short version of that is, what does it look like in 5 years?
Yeah, I think to answer that question, it's because that's going to be different for every company, and not just necessarily on your what stage of company life cycle you're at, but what's your therapeutic area. Vaccines are not quick, right? So in 5 years, there will not be a licensed product. There won't be, even in our internal lead candidates, but it should be clinical trials because it depends on which asset is in development. Some will be faster because you don't have to do efficacy trials. Those that do require, they're just more head-to-head comparisons, those are still going to require all the way through phase 3. But those vaccines that do require an efficacy trial, you're likely in a 3 to 6 year range just for that phase 3 trial. So 5 years from now, we should have clinical assets in phase 1, phase 2, and then the next phase would be usually Moderna is the exception to the rule. Most vaccine companies partner very early or right around phase 1 and then let big pharma take it. So that's kind of the hope is in 5 years, whether it be AstraZeneca, pharma A, B, C, clinical stage assets in phase 1-2 that they're developing.
Was there any part of writing the SBIR grants that helped inform you and teach you how to run a business? Because I know SBIRs have a commercialization plan associated with them, but were they more still like an academic exercise?
So I'll repeat that as, does the process of writing the SBIR, particularly the commercialization component, set you up at all for kind of where things are headed commercially for the company?
Yeah, I want to say yes and no. Grants are a game. And if you follow the rules and work, like, you know, this is not an ethics thing, but if you know how to talk to the reviewer, right? It's not about a business. It's about getting reviewer 1, 2, and 3 for you to be better than your peers. And so, yes, I had to learn about the vaccine industry. How is CMS reimbursed for vaccines? Who is the largest payer of vaccines? What are the multiple layers of payers? So yes, I got smarter in a business sense around the industry of vaccines, but I would say the day-to-day running the ship was just trial and error. Grants were helpful, but there's nothing substituting just failing early and often, but then asking for a lot of help, and I got that along the way.
So you had mentioned that the previous chemical technology was like 2.5 years to produce the vaccine. What is the timeline for your technology?
And so I'll just repeat it as sort of, you know, so we know that you're faster, but what is the timeline of the production using the Omniose technology?
Yeah, so again, we're not manufacturing, so can't give you, you know, what it would actually translate to. But what you can look at is precedent. Where is precedent and what can you extrapolate from there? So as an example, going from Prevnar to Prevnar 13, so Prevnar was a 7-valent vaccine originally licensed in 2000. Prevnar 13 comes out multiple years later. You're talking 5 to 10 years between that additional incremental gain from 7 serotypes to 13. Janssen, J&J, they actually have the most clinically advanced bioconjugate vaccine that spun out of Mario's first technology in Switzerland. That is a 9-valent E. coli vaccine that's in phase 3. During phase 1, they started as a tetravalent 4 serotype. In less than 2 years, they reformulated to a 10 serotype and advanced that, ultimately landed on a 9-valent vaccine. So it's not an apples to apples comparison, but you're talking about almost a doubling or tripling of that formulation in less than 2 years at the clinical manufacturing level. So we also don't hope that they're faster. We do hope that they're immunologically superior because we're not augmenting that polysaccharide in any way. That will require head-to-head comparisons with a chemical conjugate that is licensed. So Group B strep is our lead internal program, and that's our value prop for Group B strep. It's not speed, it's immunological superiority because we're not modifying it, and hopefully we'll be able to do head-to-head with Pfizer in the near future.
And I'll just add on, because we skipped over it before, but Group B strep you picked, it causes 90,000 infant deaths and 45,000 stillbirths annually.
Yeah, so Group B Streptococcus A galactiae is still one of the leading causes of maternal and neonatal disease worldwide, more so in other countries where they have access to less access to healthcare. But there are no licensed vaccines. There's 2 in clinical development. We're at preclinical development, but our vaccine will cover more types of Group B strep, so it'll cover the universe actually. There's only 10 flavors or serotypes. Our vaccine, GBS10V, has all of those formulated in. And so the idea is maybe speed. Speed would be nice because Pfizer is much further ahead of us. But really the goal will be to make a clinically superior product that it protects against all flavors of Group B strep.
Great. Other questions?
Have you spoken with FDA yet about your plans? And if not, when do you plan on speaking with FDA?
Tom said, what about the conversations with FDA?
And so for our preclinical stage assets, they're too early. Our AstraZeneca targets, we have internal candidates and that we develop, Group B strep and Klebsiella, and then we have targets that are strategically adjacent to AstraZeneca's therapeutic portfolio where we develop bacterial vaccines for them. Those are further advanced and we rely on the experts, AZ, to lead those. So it's something that we're like kind of on the sidelines in a good way. So I'll have to learn and then circle back with you when our internal candidates are more closely at that stage.
So maybe I'll just ask kind of one closing question, and that is that vaccines are more controversial today than they used to be, and particularly today, the last 2 days. And kind of curious, when you run into somebody who is, has questions about why vaccines are useful, what do you say to people when you just see someone out on the street?
Yeah, so just listen. People usually have a fear or a reason, and so don't preach to them, don't talk to them. And then I did not think of this on my own. I saw it from some pediatrician. Vaccines are a seatbelt. You can get in a car accident and wear your seatbelt and still have terrible injuries or succumb to traumatic injuries. It's a seatbelt. But a lot of times if you wear your seatbelt, it's less traumatic or less severe, or there's no injury whatsoever. I would encourage us all to think about vaccines as seatbelts. Yes, sometimes you get vaccinated and still get influenza, but the disease can be significantly less severe. It's your seatbelt. So listen, don't preach, and just remind everyone that it's not an all or none. That's not what vaccines do. They're a seatbelt.
Excellent. I think that's a great place to wrap up. And I just wanted to say thank you for everyone who helped to put this event together. And I won't capture everyone's name, but Marcia and Sophia and Melissa and Amy, Molly, Mallory, and many others who've kind of helped us to pull off this first test event of this format. And thank you all for coming, and enjoy the next hour of discussion.